Hi, everyone. The naked mole rat is, well, it's a biological outlier. It lives nearly 10 times longer than other rodents of a similar size, and a recent study in Science has pinpointed a very precise molecular reason for this. So in this explainer, we're going to break down how a single protein, one that normally suppresses DNA repair in humans, has evolved in the naked mole rat to do the exact opposite, protecting its genome and promoting an exceptionally long life. We'll start by looking at the longevity puzzle that these naked mole rats present. Then, we'll dive right into the core discovery, this complete functional reversal of a protein called cGAS. After that, we'll pinpoint the exact 4 amino acids that are responsible for this switch, and then we'll unpack the molecular cascade that it triggers. We'll also see how this effect actually translates to other species, before finally considering what this all means for the future of aging research. So, the naked mole rat, Heterocephalus glaber, is a true outlier in the animal kingdom. For an animal of its size, its lifespan should be somewhere around 3 to 4 years. Instead, it can live for up to 37. This kind of incredible longevity really points to some powerful, evolved mechanisms for staving off the damage of aging. A primary driver of the aging process is the accumulation of genomic damage, and DNA double-strand breaks are particularly deleterious. The integrity of our genome is maintained by various repair pathways, and among these, homologous recombination, or HR, is a critical high-fidelity pathway. We know that defects in HR accelerate aging, which makes efficient DNA repair a crucial factor for longevity. Okay, this brings us to the protein cGAS. Now, while it's primarily known as a cytosolic DNA sensor in the innate immune system, nuclear cGAS in humans and mice actually functions as a suppressor of homologous recombination. This established role poses a really interesting question. If cGAS is a negative regulator of DNA repair, has evolution perhaps selected for an attenuation of its function in the long-lived naked mole rat to preserve genomic stability? So, to test this hypothesis, the researchers set up a direct comparison. They looked at what would happen when they introduced cGS from naked mole rats versus cGS from humans and mice into cells, and then they measured the cell's DNA repair efficiency. And the results showed a very clear functional divergence. Just as you might predict, introducing human cGAS suppressed the cell's ability to repair its DNA. But when they introduced the naked mole rat cGAS, it did the exact opposite. It significantly enhanced DNA repair capacity. And this wasn't a minor change. It was a complete functional reversal of the protein. Now, to determine if this pro-repair function was linked to its known role in innate immunity, the researchers used a catalytically inactive cGAS mutant. This mutant, which is unable to synthesize cyclic GMP-AMP, still stimulated homologous recombination. This confirms that the protein's role in promoting HR is, in fact, uncoupled from its enzymatic function as a cytosolic DNA sensor. So we have a protein that inhibits DNA repair in humans, but actively promotes it in naked mole rats. The next obvious question was, why? What exactly is different about the naked mole rat's version of this protein? A systematic process of elimination was used. First, chimeric proteins that combine domains from human and naked mole rat cGAS localized the effect to the C-terminal domain. Further division of this domain then pinpointed the C3 subdomain as the critical region. From there, a sequence comparison identified 16 candidate residues, which were then tested until site-specific mutationists isolated the precise substitutions responsible for the functional switch. After all of that painstaking work, creating these hybrids, testing subdomains, and comparing sequences, the answer was located within a remarkably small region of the protein. The entire functional reversal of cGAS, from a suppressor of DNA repair to a promoter, was ultimately traced to the substitution of just 4 specific amino acid residues. And here they are. As the table shows, 4 specific changes, like a serine instead of an aspartic acid at position 463, are what make all the difference. The proof was quite elegant. When the researchers made these 4 human-like changes to the naked mole rat protein, it lost its ability to boost repair. And even more impressively, when they put these 4 naked mole rat-like changes into the human protein, it stopped suppressing DNA repair. Okay, so now that the what was identified, the researchers had to focus on the how. How exactly do these 4 simple amino acid changes completely reshape the protein's behavior inside the cell? Well, uncovering the answer required tracing a complex chain of events, a molecular cascade that starts with these 4 substitutions and ends with enhanced DNA repair.
Here's how that cascade works. First, after DNA is damaged, the naked mole rat cGAS binds more tightly to the chromatin and just stays there longer. Why? Well, it's because those 4 amino acid changes make it less likely to be tagged with something called K48-linked ubiquitin. That's a molecular signal for removal. With fewer of these tags, a protein called P97, which normally evicts cGAS from the DNA, can't do its job as effectively. This means cGAS sticks around at the damaged site, and it acts as a scaffold that helps 2 other key repair proteins, FANCY and RAD50, come together and get to work.
A mechanism characterized in cell culture is one thing, but does it actually affect aging in a whole organism? To find that out, the researchers expressed the naked mole rat cGAS protein in 2 different model organisms, fruit flies and mice. In Drosophila, which is a species that lacks a native cGAS gene, expressing the naked mole rat cGAS conferred multiple anti-aging benefits. These included extended mean and maximum lifespan, reduced age-related gut permeability, and improved climbing performance in aged flies. In stark contrast, expressing human cGAS actually shortened lifespan. And critically, mutating those 4 key amino acids in the naked mole rat cGAS abolished its benefits, and making the corresponding changes in human cGAS reversed its detrimental effects. These pro-longevity effects were also observed in mammals. When the researchers used an adeno-associated virus to deliver naked mole rat cGAS into 17-month-old mice, it mitigated multiple physiological markers of aging. The treated mice exhibited significantly lower frailty scores, milder hair graying, and lower circulating levels of the inflammatory markers IgG and IL-6. They also showed decreased markers of cellular senescence in multiple tissues. And importantly, these beneficial effects were absent when mice were treated with a version of naked mole rat cGAS containing that 4 amino acid mutation.
So what does all this evidence add up to? Well, it reveals a remarkably potent anti-aging mechanism, one that evolution crafted by making just a few targeted modifications to a single protein. As the authors state, their research provides a molecular basis for how enhanced DNA repair contributes to longevity. It shows that evolution in the longest-lived rodent hasn't just enhanced positive DNA repair regulators, it has also reversed the function of a negative one, turning a suppressor of genome stability into a promoter. So to summarize, 4 specific amino acid substitutions in the cGAS protein of the naked mole rat reduce its ubiquitination. This prolongs its binding to damaged DNA, where it then acts as a scaffold to facilitate the assembly of the FANCY RAD50 repair complex. And this, in turn, potentiates homologous recombination, which counteracts senescence and extends health span in multiple model organisms. Ultimately, these findings provide powerful support for the idea that enhancing our own innate DNA repair systems could meaningfully slow down the aging process. The key takeaway is that nature has already run this experiment, and it suggests that finding ways to make our own cGAS behave more like the naked mole rats by targeting how it's modified and how long it stays on our DNA could be a powerful new strategy for extending human health span.